Hello.
Hello.
So I know znam da vam je vi, tako da vi ako imate što da učite i to neko samo nek snima predavanje, pa možete gledati poslije. Nego moram da završim, bio sam bolestan prošle sedmice, preprošle sam bio na putu, tako da ne mogu i ovu sedmicu dostaviti. Nadam se da je to ok, pa vi ulogujte se, stavite nekoga da snima, ja ću pričat, pa naravno ko hoće da učestvuje slobodno, nego ako imate stvarno toliko da učite. Neće li snimat neko?
Trebao bi Armin.
Ok, super. Ja ću početi odmah u 4 sata pričati s prezentacijom, pa ćemo gledati završimo što prije. Vidite prezentaciju u ovom presenter mode?
Da.
Da, vidim.
Okay, super. So I'll just go ahead and start. So we can finish as early as possible. Just going to do a kind of broad presentation. As you see, there's a lot of slides there. A lot of the slides you'll be able to use for studying. Another thing is I would like you to prepare for the live sessions we're going to have in 2-3 weeks on the 14th, I think, and 15th of December. The 14th in the afternoon and the 15th in the morning. So we're going to have the presentations again, this time with all, as I said, all the adjustments you have hopefully written down what and how to make a presentation. It goes the same like last time. I would just like you to increase the group's total presentation time to half an hour and use the same stuff as last time. So you can either choose a paper or a concept and describe it in detail, divide the tasks in between you as you see fit. And we're going to do it live in one of the rooms in the university. So prepare accordingly with PowerPoint sliders, so you can use the remote changing of slides and such things. You can also try to add some other multimedia facets to presentations if you like so. And this is going to be the second part of the presentations cycle which we have. So the same thing goes as last time. Maybe I can make it, but you can also do it proactively. Just make it. Maybe you can just use the same Excel sheet with the same groups and just write down what's the title of your presentation and what's the project, so or the paper, so I can read up a little bit on it if that's fine. If there's any further questions before, you can contact me and then I can help you. But I think last time you did already a good job with selecting the papers. So okay. So today we're gonna talk about DNA sequencing methods, and later to the end of the presentation, if we come to that point, we're gonna talk a little bit about protein analysis also, so the proteomics part of the course. So we'll just give you kind of a brief history of sequencing, Sanger method of sequencing, which I think we mentioned several times at this point, and then sequence, which is sequencing a large piece of DNA, basically. And then we'll talk about the $1,000 genome, which is basically now should be recoined a $100 genome rather than $1,000 genome. But yeah, this presentation was using quite older data. I think I did it 10 years ago, maybe. So why sequence DNA? So as you know, all genes available for an organism to use are very important tools for biologists, not just sequencing of genes, but also positioning of genes and sequences of regulatory regions. You're aware that we have roughly 20,000 coding genes in our genome, which kind of make somewhere in between 40 and 200,000 different proteins, depending on what you think the whole proteome consists of. And it's kind of a vague estimate. Let's say 40,000. Some say it's 20,000, but let's even say 20 to 200,000 proteins out of those 20,000 genes. And so new recombinant DNA constructs must be sequenced to verify construction positions of mutations and similar things. So you use it also not just as exploratory thing, but also the verification when you do your cloning or any kind of vector design. Use sequencing in order to get the readout of to see if yours transfection was effective, or if you have your fragment copied into your cells, or if you have a tag, for example. So you can look into the tag also via sequencing. The alternative way would be doing some targeted PCR, obviously, but then you obviously would need to know the sequence a priori. And some rough history of DNA sequencing, if this can be counted in such a way. So we have the kind of discovery of DNA in the late 19th century. Then we have the Avery's proposed genetic material, and then the famous Watson and Crick double helix in the 50s. Then we have the sequencing of yeast RNA in the late 1960s, and then sometimes 1970, late 1970s, early 80s, we get our now known Sanger and Gilbert sequencing methods. Both are still used to extent to this day, but I think Sanger has stayed mostly around in clinics for the several past decades. And then we have several also other milestones which we think to be important in the field of genetics. So we have M13 cloning, and then we have the automated fluorescence sequencing, and several other kind of important steps along the way, right? And then methods of sequencing, we have Sanger sequencing, which we have a primary extension chain termination method. Most popular protocols for sequencing, very adaptable, scalable to large sequencing projects. The Gilbert sequencing approach is DNA labeling and then chemically cleaved in a sequence-dependent manner. This method is not easily scaled and is rather tedious. Tedious means time-consuming, basically. And then pyrosequencing, I think at this point you should just know the terms, but not really know anything about extension by pyrophosphate monitoring. Is just a stupid approach, considering next generation sequencing today. Then for Sanger sequencing, what would you need? You would need a single-stranded DNA template, a primer of DNA synthesis, DNA polymerase, and then the deoxynucleoside triphosphates to do the annealing. And basically, you would kind of split up your DNA into single-stranded parts and then realign it, and then read the realigned parts with the differently labeled nucleotides, so they glow basically, and then you have a readout of what the exact sequence thinks, because it just anneals another oligo basically, which is supposed to be on the complementing side, so it can read exactly the sequence to 100% certainty. It's very kind of just reading what's going on. So oligonucleotide primers can be synthesized, or oligos for short, right? When I say oligos, I mean this can be synthesized by several chemical steps. And now we have companies like Eurofins. I think they're the world leader in production of oligos. You can just call them and put in a sequence and they ship it to you in several days. And it's very cheap. It's like €5 or something like that. It's extremely cheap. And then the oligos must be complementary to DNA flanking sequencing regions, which is kind of something you should know from, I think, your genetics course or microbiology when you do PCR. And oligos are usually 15 to 30 nucleotides in length. So when you design it, you should be kind of aware of that repetitive regions do not repeat in comparison to similar genes, right? And also you need to be aware of the CG content because that then depends temperatures of annealing and your, for example, PCR cycle, and you need to take that into consideration. So there's several things you need to consider when designing oligos, but basically you can do it just on web pages now, where they tell you immediately what the content is and if you should redesign if it's too similar to something else or whatever. So DNA templates for sequencing, single-stranded DNA is isolated from recombinant M13 bacteriophage containing DNA of interest, double-stranded DNA that has been denatured, non-denatured double-stranded DNA, so in cycle sequencing. And this is just a brief kind of way of obtaining single-stranded DNA from a double-stranded source with magnets. It's kind of just the way how you denaturate double-stranded DNA with beads. So just pull them apart, basically. So, and DNA polymerases, which do the chemistry of it, the annealing of our oligos, should be highly, of our nucleotides, should be highly processive and incorporate the NTPs efficiently, should lack exonuclear selectivity, and be terminally stable, right, for cycle sequencing or even doing a PCR. So up to 95 degrees, obviously. And then Sanger sequencing, the basic methods. So you have your single-stranded DNA, and then you have the annealed primer, and then it basically reads from one side to the other the stuff it glues onto the single-stranded strand via laser. It's actually quite ingenious, quite straightforward. And then Sanger, this is just a schematic show of the ATCs and Gs, and then how the growing strand develops by adding your DNTPs and then obviously your DNA polymerase and all this stuff. And then the DNA is broken up and you have one single strand, and then the temperature obviously then degrades the DNA, but then you have the DNA polymerase and the DNTPs, which are then adding the newly formed double-stranded strands together. So then the single strands, which are kind of made from the temperature, and then they're realigned through the chemical process. And this is again how the basic method looks. So the ATP is the reaction. Anywhere there is a T in the template end, occasionally ADD will be added to the growing strand, and then you just read it as it goes. How to visualize DNA's fragment. So you can do it by radioactivity, which was used to be done 50 years ago or something. But then I think the more easier way, even though also quite toxic, if you use a tedium dibromide, for example, is with fluorescence. So DDNTP's chemical synthesized contain fluors. Each DDNTP is fluorescence at a different wavelength, allowing identification. So and then you would run a gel, for example, and you would see polychrome yellow crevices, good resolution to fragment different by a single DNP. Slab gels as previously described in the book, and then capillary gels, which require only a small amount of sample to be loaded. And then you could also read out your nucleotides like this, and to verify and to see what's going on, which sequence is where. And then this is then cycle sequencing. Denaturation occurs during temperature cycles, similar to your PCR, basically. And in this page you have animations, and this would be what they call an automated sequencer, which is basically just Sanger sequencing machine, which is standardized. And then we can actually skip this. This is the shotgun method. I'm just here going through the different methods, so you know a little bit more detail from the presentation. There have been many approaches into sequencing, and it's just that we recently agreed on that we use NGS and Sanger sequencing rarely, but also those two are 95% of sequencing nowadays. And then this is a shotgun. This is how the Human Genome Project was sequenced with this shotgun technique using M3 bacteriophages, and it was a tedious project, taking decades actually to finish. And I think this is the original paper, right? Genome sequences of the human malaria parasite. No, this is a different paper, but it was done by the Sanger Institute as well. Sequencing strategy, a whole chromosome shotgun sequencing, which was used to determine the clone identity here in this paper, which was referenced previously. And this is also just going through some steps of the paper, which is just describing more details, but you really don't need to bother with this too much. So, and this is the $1,000 genome, which is now the $100 genome, and I think last year it was achieved in China, so you can commercially sequence your genome below $100, and it costs roughly, I think, $95 to do in China. I don't know if they have any bulk discounts, but in general, we're already there at the $100 genome. And comparing with the thousands and millions which it took to do it previously. Yeah, this we can also skip. This is just basically talking again about different approaches in sequencing, and this is different extension with individual DNTPs, which gives a readout, and you can imagine all of those techniques basically as being an approach of that's basically big PCRs, right? So you have your DNA of interest and a template, and then you denature that and try to add glowing DNTPs on it, so they can be read by a laser in a different pattern, or eventually you can read them out on a gel, as mentioned previously, and then you see all the different ATs, T's and G's in the gel, and then read the sequence from the gels, which you can imagine is super time consuming and tedious, and I just can imagine how boring it was to do all this stuff like 50 years ago. But now this is completely redundant, right? So you don't use this approach any longer. And yeah, this is the DNA library preparation by titration, making it kind of very slow, 25 million bases in about 4 hours. You can imagine how long it would take then to sequence a whole genome. Usually now it's, I think, 7 or 8 hours to do a whole next generation sequencing human genome. And this is how a readout would look like in those approaches like Sanger sequencing. Height of the peak indicates the number of DNTPs added, and so this sequence then reads TT, 4Gs, T, GC, A, GTT, and this is basically the sequence of your gene of interest, and this is how a graph would look in those outputs. Completely different to the stuff we were talking about previously with the FASTQ files and raw sequencing data. So you can imagine this is not very high throughput, and doing thousands and thousands of patients becomes easily unobtainable in this way, because it's just too tedious. It would take years to do that. And then just kind of having a small introduction to so that you have all this stuff in one place. Have bioinformatics as a tool to analyzing the stuff we mentioned previously. So making biological sense of DNA sequences, We have obviously our online databases, which are quite old in this presentation, but nevertheless they're still active. Most of them, they're much updated because this presentation was old when I was doing it, right? So, and then we have NCBI and we have BLAST, which you all know from your studies, using BLAST for sequence analysis and so on. So making sense of sequences, first we want to see the genes and understanding if this gene is protein coding, where are the open reading frames or ORFs, right? This is the shortcut. What are the open reading frames most similar to? What is the function, structure, evolution, and history? Then we want to see RNA, which similarly, in order to analyze RNA, you need to make cDNA out of your RNA and then be able to analyze that cDNA out of the RNA. And then we have non-genes like regulation, promoters, and transcription factor binding sites. And then we have obviously replication, repair, and segregation, DNA packing, chromatin unfolding, and those kind of things. And this is a classical readout which you would have from a Sanger sequencing machine, which is indicating those colorful peaks, which are different excitations of the spectra of light, given a different DNT pace in a given position. On the left side, you see how it should look, and the right side, how it shouldn't look. So the right side, you can't really read anything there. It's very bad. And you get this NNs, which basically tell you that the read could not be done at that place. And then you have like a sequence, could this encode a protein? And then you look at the open reading frames. This is all done in BLAST, right, and NCBI. This is like a usual sequence would look. And then you can go and look into the open reading frames and then where the potential starts, the ATCGs, and where the stop codons, which reading frame is correct. And if you have a frame shift to see what's going on, how it affects eventually your protein. Because even with the frame shift, you can obtain a functional protein, just different in size, for example, which is quite often the case. And then you have issues with the antibodies. So it's also good to see when you have transformed cells and look into this part with the open reading frames to see where eventually something has shifted. So you can know when you analyze with your antibodies, with your Western blots, for example, to see if your antibody will specifically bind or not bind to a different site. So one should be cautioned when identifying open reading frames. Not all genes initiate with a TCNG, particularly in certain microbes. What is the shortest possible length of open reading frame? 50 amino acid, 25. Cutoff is somewhat arbitrary. Nobody really knows. In eukaryotes, those frames can be difficult to identify because of introns, and you know what introns are, also parts of the gene, but do not really do much in composition of the protein. Are there other sequences surrounding the ORF that indicate that it might be functional? Promoter sequence of RNA polymerase binding, and then you have the Shine-Lugano sequence for ribosome binding. What is the function of the sequence gene? So we can have a look and analyze it. So the classical methods, so mutate genes, characterize phenotype for clues to function, which would be genetics, and then the purified protein product characterize in vitro, which would be the biochemistry approach. And I think nowadays those approaches are synergized into one. Basically, there is no big difference between the two. People who do one do the other two. Comparison to previously characterized genes sequence that have high sequence similarity usually have similar functions, which is logical because they would make a similar protein, and then depending on the structure and folding of the protein, the functions could slightly deviate or be different, but nevertheless different enough to do different works in the cell in a single cascade, for example. If your gene has been previously characterized by someone else, you want to know right away. And this is just that's why we have the NCBA database where we can have a look in all this stuff. So we have PubMed, as you know, all that sets, BLAST, books, text browser structures. So everything is there at one place. So you don't really need to worry too much in those nowadays. You can just literally type the gene name and even the sequence if you have obtained a newly sequence, which you're not sure that it's obtained before. And I think yesterday published the unknown, like the genome. You have the unknown for genes that we have still no idea what's going on and what they do. It's a kind of interesting paper. You can have a look into it. It's called the unknown genome. It's very cool, actually. I mean, cool. For normal people, obviously not, but scientists, cool, right? So you can have a look and see that actually it's quite amazing how much we still do not know about the genomes 20 years on of the Human Genome Sequencing Project. And something I think you have talked about ad nauseum at your university. So what does BLAST do? So it searches chosen sequencing databases and identifies sequences with similarity to test sequence, ranks similar sequences by degree of homology, which is the famous e-value, and illustrates alignment between test sequence and similar sequence. So the principle, basically, two homologous sequences derived from the same ancestral sequence will have at least some identical amino acid residues. So you can imagine, for example, insulin is quite conserved, right? But it's kind of also different. So you would see that dog insulin and human insulin are similar enough that people can work and live on dog insulin, as they did actually. Quite until recently, it was extracted, I think, from dogs or from some of those raccoons or who the fuck knows what animal, but it was done by killing thousands and thousands of animals and extracting out of the pancreas insulin, which is quite conserved, but when you look at the sequence, you would still see differences, obviously not big enough that it doesn't work in human bodies, but still there. So a fraction of identical amino acids is called percent identity. So similar amino acids, some amino acids have similar physical chemical properties and more likely to substitute for each other. These give specific similarity scores and alignments, right? So gaps in a similar or homologous sequence are rare and are given a penalty scores. This is for BLAST. Homology of proteins, homology, similarity of biological structure, physiology, development, and evolution based on genetic inheritance. Homologous proteins, statistically similar sequence, therefore similar function, often but not always. Alignment of TBF and TFFA sequences. So high sequence similarity correlate to functional similarity. And then, there's kind of this 8 different kinds of proteins in your body. You have the hormone proteins, the signaling proteins, the transport proteins, the hormone proteins, whatever, right? So, and depending on the class of protein, then this score would be differently affecting the homologous approach of one species to the other, for example. But obviously, the more evolutionary close you go, the more similar the protein structure anyhow is, or the sequence structure anyhow is. So if you have 40 to 20% identity, fault can be predicted by similarity, but precise function cannot be predicted. This is the blue. So programs available for BLAST searches, protein sequences. So you compare an amino acid query sequence against a protein sequence database. Then you can compare a protein query against a nucleotide sequence database translated in all reading frames. Then you obviously that for the DNA sequences compares a nucleotide query sequence against a nucleotide sequence database, compares a nucleotide query, and so on, not to read this whole through. And then the last considers all possible combinations of messages, mismatches, and gaps in any given alignment, gives the best highest score alignment for sequences. 3 scores, percent identity, similarity score, and the famous C-value. Probability the two sequences will have the similarity they have by chance, lower number, higher probability of evolutionary homology, high probability of similar function. So it's the same when the virus came out, the corona stuff. That's how we would basically determine where it comes from, and given the structure of the S protein or the E protein sequence, you could then deduce some function in comparison to other species of coronaviruses, for example, and it was actually done quite early. And again, this is good for you to know because you finished this university, but in general, I don't think you're ever going to use this to explain to someone. So the E will represent the chance that the similarity is random and therefore insignificant. Essentially, the E value describes the random background noise that exists for matches between sequences. For example, an E value of 1 assigned to a hit can be interpreted as meaning that in a database of the current size, one might expect to see one match with a similar score simply by chance. You can change the expected value threshold in most main blast search pages. When the expected value is increased from the default value of 10, a larger list with more low scoring hits can be reported, right? So this is from the last tutorial. Although hits with e-values much higher than 0.1 are unlikely to reflect true sequence relatives, it is useful to examine hits with lower significance for short regions of similarity. In the absence of longer similarities, these short regions may allow the tentative assignment of biochemical activity of the open reading frame. The significance of any such regions must be assessed on a case-to-case basis. This is just how I would define this stuff. And then you can see, obviously, the most important part is the relationship between the E function and but the E value and the function, right? So single domain proteins, multi-domain proteins, you see there's a slight difference. Different relationship of those two, right? So E value greater than 10 to the minus 10, similar structure, but possibly different functions. And this is just what does the sequence do? Queue up blasts, and I won't bother with this. But you have the presentation, so you can technically go and do it by yourself if you're really interested in this. But I don't really think you need to do this. Again, this is just for you to have this presentation at one place with all this stuff, which is kind of the guidelines for this course, right? Because I'm teaching mostly DNA sequencing data analysis, but the course is genomics and proteomics, right? So having this one presentation, so we can cover this part also in one go, talking about those things in synchronization, and then we can then continue from tomorrow doing our computational work. So I wanted to get this out of the way as soon as possible. So yeah, let's go further. Okay. So this is then the kind of next part of this presentation is making biological sequences. New DNA sequences are analyzed for open reading frames. Any DNA or protein sequence can then be compared to all the other sequences in a database, which is the whole genome or cross species, and identify similarities between individuals or species. There's much more a great diversity of program and databases are available on NCBI. So massively parallel measurements of gene expression, so-called microarrays, defining the transcriptome, the northern blot revisited. Nobody uses northern blot anymore. Detecting expression of many genes, array, a typical array experiment. What do we do with all this data? Yeah, this is a paper from 1999, so you can imagine that we now know what to do with all of this data. We store it in clouds and analyze it on servers at this point. So then we have this maybe interesting part that people still think very much in the sense that we have DNA, RNA, protein, but there is so much in between, right? So in between the genome and the transcriptome and the proteome, and that's why you have genomics, proteomics, and transcriptomics, right, to describe all those things. We now know a lot of the transcriptome, which is a lot of cell-dependent, environment-dependent, as you know. And the proteome, I think, is largely still a mystery because, as I told you from the beginning, we estimate somewhere, let's say, 200 to 200,000 different functional proteins in the human body, which is not a small margin of error. It's like 10 times difference, right? It just depends on what context you ask the question. So microarrays are a very interesting thing, actually. I use them by myself for DNA. They're actually quite time useful, and they help you not mess up things with those preset probes, right, for expression or even polymorphisms, right? You don't need to do data interpretation as you would do with DNA sequencing, or the variant calling. The variants are already there, so the microarray just does it all for you basically. And it's actually older technology in DNA sequencing. It's because it's all pre-hybridized probes, and it's quite straightforward to use. Just load DNA on a chip and put it into the machine, and you get the readouts immediately. But then you can use it also obviously for DNA expression, and then you had to study different transcription at different times. But then this is also the redundant way, because as you can imagine, the RNA which you get from those cells is the bulk RNA, or mostly ribosomal RNA, and that doesn't really tell you much what's going on actively in a cell. And it's an average from all the cells you extract RNA. So I think at this point, bulk RNA becomes a really redundant technique in comparing to single-cell RNA sequencing, because single cell is now the gold standard. You don't really trust bulk RNA data, because now there are actually ways also deconvoluting the data where you can have bulk RNA and then reverse engineering, so to say, and then see from what cells it comes. But yeah, it's just stupid nowadays if you have 10x genomics readily available at most advanced labs. So there's no really need to do this. But we have already generated so much bulk RNA data over the last 15 years that deconvoluting might seem also a great way of analyzing the already existing data. So, and then we have the famous northern blot, which you study northern, southern, western blot. It's very funny naming of blotting techniques and gels, which is immobilize mRNA on membrane, detect specific sequences by hybridization with one labeled probe, requires separate blotting for each probe, which is just takes a lot of time. And then DNA microarray immobilize many probes, thousands in an ordered array, hybridize base pair with labeled mRNA or cDNA. And you can do a lot of identification of open reading frames and PCR of each of those frames. Each PCR product to a solid support. This is how the array probe works basically. Chemically synthesizes or specific oligonucleotide probes directly on microchip Affymetrix, which is the most famous chip producer and most famous platform. So if you do any of those 23andMe tests or MyHeritage and stuff, they all use Affymetrix. And if you use it, then if somebody has actually done that and you can get the data, I can show you how to analyze your own data and get some cooler information out of that stuff if anybody has done it. And it's great. I think it's quite cheap. It's like 30 euros to do or something now. And this is how those chips were built. Chip design with certain parameters already in mind. Obviously not the most hypothesis-free way of generating a set. But nonetheless, this is just the workflow of all these old approaches. Let me see, do we need to talk about this? So and this is how you would see your microarray data expression. So it would be a color way with green, red, and yellow. Depending on color, you would see that it's red. There is more abundant mRNA for that gene, right? So like you would assume there's more transcription going on, and then the yellow would be kind of in medium level, and then the green, there's not much going on basically. And so how do you can interpret it? The data, you look at patterns of expression, and you get those heat maps if you know how they look, organized data by hierarchical clustering, profiling to find patterns, displayed graphically, and this is one of those graphs, how they look depending on different, obviously, cell cycle stages of different genes and how they're expressed during different stages of the cell cycle, with high mRNA levels being red as mentioned, and then green we would have a low mRNA levels. And this is just a representation of a yeast cell cycle regulation with CDC28, LU, CDC15 and alpha. And then we have the minimum information about the micro experiments, so experimental design, the set of hybridization experiments as a whole, array design, each array used each element, sample use, extract preparation, hybridization, measurement, images, normalization controls, and so on. And this is then giving the famous answer about proteomics of this course. So looking into the protein and how to analyze protein, as you might know, there are several ways of looking into this. So we have which proteins are present and when, what are the proteins doing, what interacts with what, or protein-to-protein interaction, and then protein to DNA interaction, chromatin immunoprecipitation, which is kind of a cool technique where you can just kind of pull down chromatin and look what's going, because you always imagine DNA as just A, T, C's and G's, right? But those are not in a vacuum, right? They're together with a lot of structural proteins, this nucleosome stuff, and then adjacent proteins. Adjacent proteins. So chromatin is as you know, the whole structure of DNA and proteins together. It's not just the DNA. You never have only DNA by itself, except until you purify it in a column or something, or precipitated. So we had the classical methods of proteomics, so using antibody for specific assays, measuring changes in protein levels with changing environment in different tissues. You could use stuff like ELISA and mass spectrometry and markers as well. And then we have this famous 2D gel separation, separate proteins in a given organism by migration factors. So you have Western blot, for example, where heavier proteins would stay on the top of the gel and lighter proteins would go faster given a current. And then you would have your lower proteins and smaller proteins down on the bottom, and on the top you would have your heavier, bigger proteins. And using an antibody to try to see on this gel if there is this protein present, and then you go in a dark room or use an imager to see the bands on the gel. So then we have mass spectrometry. We're separating individual proteins from cells by charge and mass. Individual proteins can be identified. You have also protein crystallography and all this stuff to get the structures. So it's kind of an interesting world in proteins. And you always have to think, like, it's all fine and neat with DNA and RNA, like they're cool and everything. But proteins are carriers and doers of function, right? So you don't really care about the mRNA levels that much. It was used to think that mRNA levels are good expression profile, good proxy for proteins, but there is so much stuff going on after mRNA synthesized. We don't really know how those metrics even correlate. Some even think it's complete garbage to use mRNA as a proxy for protein, but that's up for debate. And microarray isolates things that bind to proteins. And then this is just separating proteins on different levels. With high pH, the protein is negatively charged. At low pH, the protein is positively charged, and then goes to its gradient. This technique is usually done on a long, narrow gel. And then this is what you get. It's a kind of pH-based separation. And the mass spectrometry for identifying proteins in a mixture. So you kind of hit it with different rays, and then liquid chromatography, and then the mass spectrometry software proposes data, and then you get the scatter plot. And depending on the mass of the different molecules, you get a plot which tells you exactly what's in your solution. And then given on the size, you know what proteins, what size your protein should be, and then you can look for the presence of that protein. Defining protein functions. So we have different, so you can do knockouts, for example, where you can knock out a certain gene making a protein, and then you observe what happens with that cell culture if you knocked out the gene. You can purify proteins from cell and characterize their structure and function in vitro by using crystallography and stuff. So there's several different ways of doing. Now it's also redundant. It's not time intensive any longer to do a knockout gene. You just do it with CRISPR, which is basically 72 hours, and you knocked out the gene perfectly precise with like a one base pair, a standard deviation, which is nothing. So you get really, really good results with CRISPR. Protein DNA interaction, as we said, so DNA and protein, you can use several methods to pull down your chromatin, but basically fixating your DNA and proteins in a given stage with, for example, you treat it with something, you want to see how the chromatin changes on and the protein structure changes on DNA, and then you label it with something which is either magnetic or can be bound to some beads, and then you pull it down with magnets, and then you magnetic beads, and then you elute the magnetic beads with heat, and you get your protein and DNA compound completely eluted into like any kind of buffer, which is ready then to be done run on a gel. Chip is a cool technique. I used to do this in my old job, and it's kind of the very same approach I explained. So you grow cells at formaldehyde to cross-link, which means basically just making it stable so it stays like it is. Then you lyse the cells, open up the cells by lysis buffer or by sonication. And then we have uretreated proteins using specific antibodies to that protein, immunoprecipitate, and determine the presence of DNA by PCR. So this is, and you can do it in massive parallel chip. And it's called chip-seq, so you can call it chip sequencing, or you can sequence the gene of interest in a highly parallel way. And this is the readouts which you get, so protein fused with 6 histidine tags immobilized by Ni++ matrix, and then you see it's just presence is red, absence is red, and presence is green. And yeah, that's basically the presentation. We had 45 minutes to the dot. Good. So I hope this is recorded. So we now have this presentation done with the proteomics part, and we will not mention this too much any longer. We'll just continue with tomorrow with computer stuff, with the sequencing, data analysis, and then as I don't know if everybody was here when I started talking in the beginning about the presentations, which are gonna be done when I'm there on the 14th and 15th. Please put your names, or put the papers which you want on that Excel sheet, and tell me the name of the paper which you're gonna do, so I can read it in beforehand. Before we can, the presentation is gonna be live. I'm gonna try to find a nice big room where you can do it, and we can either split it in two, so 2 groups one day, 2 groups other day, or we can do it either the whole day on Thursday, which is quite stupid maybe because I'll just allow it's the same. The paper or the presentation should focus on sequencing, right? So preferably it would be data analysis. So how the stuff we talked about, the FASTQ files, how you generate them, and some quality control, but also a disease is fine, a complex disease understanding of using sequencing, monogenic disease, all the stuff which is also kind of mentioned in the which was presented in the midterm papers question. So all that stuff, if that answers your question. Yeah, but just yeah, good. So yes, the sooner you decide on a paper, you can just put it in there, so I can also prepare. But I'll give you a deadline. So now it's the 22nd. So 1 week, 2 weeks, 3 weeks, right? So I'm in 3 weeks there. So by the 29th, no, you have this week's exam, right? Let's say by the 4th, you should all have written down and told me what's going to be in your presentation so I can read it through. I hope that's good. Any questions?
No questions.
Okay, good. Some of you, I can't remember, maybe answered the email. Send me an email about doing some stuff, bachelor thesis or some things, if I remember. Who was it? Is there? Should I do anything there? I didn't catch after the email because I got sick and everything. I can't remember who sent me the email. None of you?
Ines wrote in chat.
Yeah, possibly. I can't remember.
Zašto mail? Zašto mail?
Pa da budem supervizor za bachelor.
A ne, nisam.
To sam se ja javio preko Vibera, nisam vam mail slao.
Pa izvini, meni, ne znam ti ja točno, že sam se javio, nego jesi ti razgovarao šta sa Adnom, jel treba nešto pisati, neki prijedlog, kako to funkcioniš, treba li šta
Pa
pomoći?
to ćemo, kad dođe vrijeme za to, sjest neki meet napraviti, možemo i 14. 15. se dogovoriti, pa...
Ja, pa odlično, tamo sam ti to htio predložiti da budem tu, da sjednemo i da se dogovorimo. Dobro, to je to za danas, završili smo s ovim Protomics, ja to više neću spominjati, sad nastavljamo samo rad DNA sequencing i genomics, ja sam to obavio sad i sutra opet oko 4 i to je to onda. I nadam se tamo od sljedeće sedmice da punom parom, nadam se da vam dobro ide sa ispitima i da vam ovo što sam ja poslao nije previše teško, ne bi trebalo biti. Eto, to je to.
Bye.
Dogovoreno, čao, čujemo se, bye bye.
Bye, prijatno.
Bye bye.